welcome to the second of 3 Loeb lectures by our distinguished visitor, Cristina Marchetti, who is indeed the distinguished professor of physics at UCSB. I guess the other ones are undistinguished.
No, there are many distinguished.
There are many distinguished, of course, as there are at many of our institutions. So this, I guess, will be a little bit more informal. Those of you who didn't hear her beautiful talk on Monday are in for a treat. And she's going to tell us today about the dynamics of active fluid interfaces. And I guess it's okay if people ask questions.
Yes.
I guess the request is that you just repeat the question.
Okay. And if I forget, you can remind me.
Thank you.
Okay. Well, thank you, David. So yes, so yesterday I told you a little bit about these active pneumatic fluids and the self-sustained flows they exhibit. And what I want to do today is tell you about the interfaces between active pneumatics and passive fluids. And this is work that was, there's going to be quite a bit of experimental work here, because that's work really that was motivated by recent experiments done in the group of my colleague Zvonimir Dojic at UCSB, and especially done by a couple of really great postdocs, Itamar Colvin and Alex Tayar, and the student Ray Atkins, and an undergraduate Sven Witthaus that has now moved to the University of Chicago. And I'll tell you also about theoretical work we did to try to understand these experiments, and this was done by former postdoc Jihong Yu, Fernando Caballero, and Parth Gulati. And essentially what I want to tell you about today is how activity essentially modifies interfacial phenomena, such as the kinetics of liquid-liquid phase separation, the structure and dynamics of fluid-fluid interfaces, and also wetting. I probably won't tell you about wetting though, but if somebody asks me at the end, I can tell you about that. So in some sense, I hope to convince you that activity can be used as a new handle for controlling the properties of liquid-liquid interfaces. Let's see. These are today. It doesn't work. Okay. No, still not. Okay, whatever. All right, so I'll have to stand here then today. So as you know, immiscible fluids such as oil and water, shown up there, phase separate, creating these soft fluid interfaces, and the liquid of the physics of liquid phase separation has many applications, from cooking, making salad dressing, to material science and engineering for the formation of a variety of soft material, vesicle, metal structure, and so on. And most recently in biology, where it drives the condensation into these membrane-free organelles, like the ones as shown here, which are something called P granules in the C. elegans embryo that are implicated actually with determining cell fate. This kind of soft interfaces, I don't know why this doesn't work. Oh, now it works. Okay. This kind of soft interfaces can be broken up by applying external forces. So for instance, by shaking the container, you break up the interface, and then this energy input from the outside break up the interface, and then actually you restart the process of phase separation. And we're going to see how activity, which energy injected from, here energy is injected from the large scale to the small scale. We want to ask how activity, which is energy injected from the small scale to the large scale, modifies this process. I should say that yesterday I told you about a phenomenon that I call motility-induced phase separation, which is the phenomenon where active processes, in this case motility, induce phase separation in a system that does not phase separate where passive. Today, I want to tell you about activity modifies the phase separation of systems that do phase separate when passive. So it's going to be a little bit different in that sense. Just as a quick reminder about some things I said yesterday about active fluids. So active fluids are fluids made of entities that consume energy to drive themselves, to exert forces on their environments. And this energy input at the particle scale cascades to large-scale dynamics, creating, for instance, this is a laminar flow of a monolayer of epithelial cell in a channel. And this is the type of turbulent-like motion of the mixture of cytoskeletal filaments, microtubules, and motor protein that actually we saw yesterday. These are both two-dimensional type of flows. But in addition to this kind of bulk active fluids, there are many situations in which active fluids coexist with passive ones and create interfaces. So here is actually the interface between motile and non-motile bacteria in experiments by Ellison Pattison, actually at Syracuse University. This is an interface of a growing colony of bacteria. This is just an image. And this is actually a non-living system. This is a fluid of colloidal chiral rollers of magnetic particles that rotate continuously. And they create, these are experiments by William Irvine at the University of Chicago, and they create this interface where you also very clearly see traveling waves at the interface. So this is just sort of some kind of a general motivation to kind of ask the questions that I already raised of how this activity can actually modify interfaces and liquid-liquid phase separation.
Cristina, could I ask?
Yes, please.
At the bottom right, in Irvine experiment, we're looking at a two-dimensional sort of top view of a monolayer of these rollers, and they feel.
This is actually, no, I'm not going to talk about this system. This was just for kind of, this is actually, I believe it's a very thick layer. So it's actually a cut through a system, which is not that thin. Yeah. Yeah, I think so. Okay. Yeah. And then you see bubbles also that are, yeah.
We're supposed to notice not only fluctuation, but there's a chirality going from left to right.
Yes, there's chirality because these are chiral particles. So this is actually, they're rollers, they're particles that are all driven by a magnetic field. So that is not surprising to be chirality at large scale, but because there is chirality at small scale. But I'm not really going to talk about this, it was just as a motivation, really. All right, so we want to look at the effect of activity on these interfaces. And the kind of system I'm talking about are just kind of to remind you about what I said yesterday, they are sort of inspired by biology. There are these suspensions of microtubules and clusters of kinesin, motor protein, that convert chemical energy into mechanical work to drive these biofilaments to move relative to each other. And in the cell, they actually rearrange the cell's cytoskeleton and the so-called mitotic spindle. So this is actually a natural cell undergoing cell division. And what you're seeing here are these microtubules that separate the chromosomes during cell division. And the kind of fluid I'll be studying that I already showed an image of and I showed you yesterday are actually in vitro reconstituted system made of these ingredients, essentially, the same kind of ingredients. And yesterday I only show you, but I just want to show you a little bit more in detail what's happening in this system. So there are two-dimensional layers at an oil-water interface. And here is a video from the Dogic lab at low density, where you very clearly see these bundles of microtubules that extend, break, and perhaps reconnect, and in doing so, exert forces on their environment. And at higher density, these forces drive these turbulent-like flows. And actually today, I'll be dealing with systems that are at a lower density of microtubules than yesterday, so that in the absence of activity, these systems are isotropic, they're not nematic. Although, as you can actually see already here, activity sort of builds up quite a bit of local nematic order in the system, and that will be important. Yes. Say it again. Oh, so these are the scale. There is no scale here, but it's probably it will be several minutes, I think. So these are pretty fast kind of motion. Okay, and this is also something I mentioned yesterday, but I just want to make sure the talk is self-contained. So these flows are driven by these active stresses, which are determined by the dipolar forces the extending microtubule bundle exert on their surrounding. And we can actually write the net force on the fluid element as a sum of all the force dipole. There is no net force, so the first non-zero term is the gradient of a stress, which is what we call the active stress, proportional to the order parameter of the nematic and to this quantity we call activity. And again, we are interested here in systems that are extensile, that is, the dipolar forces point out, and this activity parameter will be negative. Okay, so what do we do with this? Now, in order to study interfaces, the first challenge was to actually construct a system in the lab that can be controlled and that does phase separate. So the way the Dogic lab did this is to take a polymer mixture that phase separates in equilibrium, and precisely they took a mixture of PEG and dextran. And one of the reasons for choosing this mixture, as we'll see, is that it phase separates and it has a very low surface tension. In fact, about 6 orders of magnitude lower than the surface tension of water, and comparable in magnitude to the kind of forces these generated by the kinesin motor in the active system. Then they combine this with this microtubule-based active fluid. And the microtubule partitioned completely into the dextran-rich phase, which is the light color phases in here and all the images I will show later on. And so now you have a mixture of the dextran plus the microtubule, that's the active phase, and the PEG, which is the passive phase. And as I mentioned, this system has a very low surface tension, which will be important here because that way activity can actually drive interface fluctuations.
So Cristina, these are both polymers, if I understand correctly?
Yes.
And dextran is not branched, it's just a linear polymer, and they have comparable polymerization index?
I think so.
And then what renders the dextran active?
So they take this mixture of PEG and dextran, they add this, they mix it up with a microtubule kinesin active fluid.
Oh,
and they can demonstrate by labeling with different colors fluorescently the dextran and the PEG and the microtubules. They can actually demonstrate that the microtubule completely segregate to the dextran phase. So dextran plus microtubule is the active part and the PEG is the passive part. And the system phase separates. And so then to study the interface, they actually put the whole thing in a vertical cell to thin glass walls separated by a small gap. They centrifugate the whole thing. The active fluid goes to the bottom because it's heavier. And they can see this interface, which exhibits a very large fluctuation. Here the scale is millimeters. So these are actually practically visible by eye. And this is clearly an active effect because if you were to calculate with using the surface tension and temper of the system and temperature, the RMS fluctuation of the height that you would expect if it was driven by thermal fluctuation in this PEG dextran mixture, you would actually find that it's of order of 100 nanometers. And so clearly the fluctuations here are much, much bigger.
Can you say, can you rule out the presence of gradients in certain action, which might also be possible to drive?
I don't think so. I don't know. I don't know if you can rule them out. No, I don't know.
The surface tension magnitude itself is very small, but then you have gradients in large scale, potentially large scale.
I guess so, but I mean, it's certainly okay. It's certainly true that you can actually tune the activity in the experimental system by changing the concentration of kinesin, and that you really see that without kinesin, the interface is completely flat. And with kinesin, the interfacial fluctuation size increases actually linearly, essentially, with the concentration of kinesin. So it really seems like it's driven by the activity, what we call the activity.
And your timestamp is seconds, or what are we saying there?
This is minutes, seconds.
Minutes.
Yes. Okay, so yes. So this whole video is about an hour, but then I think I looped it, so you can't tell. Sorry, yes. Oh, first of all, I forgot to repeat the questions, and now I'm actually not quite hearing the questions. So anyway, go ahead.
Yeah, you said all the microtubules go to dextran.
Mm-hmm.
Is it possible to have a situation where a fraction of them localize with PEG?
I don't think so. So the question is whether it's possible to have a situation where only a fraction of the microtubule localized with dextran. And I think this localization is driven by sort of depletion effects, and I don't think so. I don't have such a slide, but there is a slide where you can really see very clearly through this differential fluorescent labeling that microtubule would go. But I don't know. I don't know if you can control that or not. All these experimental questions, I don't know the answer to. Another question up there.
Rather than like a tubular layer?
No, this is a vertical layer. Okay, so they've done actually two types of experiments. This vertical layer, which is also centrifugated so that the active one goes to the bottom to study the interface. They've also done experiments in a 2D horizontal layer to study the kinetics of phase separation, and I'll show you that later. Okay, but this is, yeah, this is not, it's no longer the thin fluid at the oil-water interface. Okay. Okay, so I kind of have introduced the experimental system. What I wanted to do next is tell you about, we try to go about modeling this interfacial fluctuation with a continuum model. I will show you that this system, although overdamped, actually supports surface capillary-like waves driven by activity. And we have actually just started trying to understand the effect of active stresses on the phase separation and coarsening. So try to tell you about that. Okay, so first of all, let me just spend a minute reminding you about simplest way we describe phase separation in equilibrium. So it's through what's called known as the Cahn-Hilliard theory for a concentration field. So we have two species. This is the concentration, say the difference, the density of the two species, and it's conserved. And so its dynamics is controlled by an equation where the rate of change of phi is controlled by the chemical potential, which is determined by a free energy that is where the ground states are tuned by this parameter A. If A is positive, the system is mixed, well mixed, especially homogeneous. Free energy is a single minimum. When A goes negative, we have the double minimum, and the system demixes in region of high and low concentration of the two phases. The phase diagram in the, you can think of this A as sort of an effective reduced temperature, for instance. The phase diagram is the usual upside down parabolic type shape with a critical point, which is at A equal to 0 for a 50-50 mixture, and exhibits the region of nucleation and spinodal decomposition, and as this typical shape. Now, what we would like to do now here we have a system in which, in addition to concentration, we have to worry about the dynamics of the nematic order parameter field and the flows that are driven by gradients in the active order parameter field. So to this, we would like to add the coupling to liquid crystalline degrees of freedom and to active flows. So to do that, we go back to something I also mentioned yesterday. First of all, remind you again how we describe the hydrodynamics of bulk active liquid crystals in terms of a flow velocity and of our order parameter, which is defined with a different factor of 2 here, sorry, but it's the same kind of order parameter. The flow velocity, because we are thinking about this cell where the system is between two glass walls. So again, we have actually friction. So this is just a Stokes equation for force balance. And the forces are frictional forces, viscous stresses, pressure gradient. And then again, we have gradients of active and elastic stresses. and in particular, the active stresses, which are proportional to the Q, gradients of active stresses generate the flows that we see in this system. The order parameter field, its dynamics is controlled by relaxation. And notice that here I only included the linear term here because I'm assuming that I am in the passive case, the system is isotropic. And so I don't need the cubic term that one would get if the system was nematic. Yes.
Say a little about the constant density assumption in this model, because like this makes sense to me for the 2D oil-water interface flow, but like in these other systems, I don't fully understand.
Well, it's true there are much bigger density variations in this system, and we are actually trying to incorporate that density variations right now. We are finding that does not make a big difference. But the first thing we did was to assume again a constant density. So that's. And the one thing I want to point out is that this coupling to flows that I also had yesterday, obviously, in particular this term, the coupling to velocity gradients, actually tells you that shear flows generated by the active stress actually can induce local nematic order. And that's why in the kind of videos I showed you before, you actually see a system which is, of course, isotropic on average, but there is quite a bit of local nematic order. And you can actually show that in some sense fairly simply because you have this divergence of Q here that in the limit in which certainly viscosity is high, becomes of order gradients to the zero, and so actually changes the sign of this relaxation term. So induces local nematic order.
Make sure I understand the first couple terms in the bottom there. ST is a symmetric.
Oh, symmetric and traceless, because Q is traceless. Traceless symmetric part of the gradient. So this is supposed to be a tensor kind of product, right? And so this will, yeah, and this is the vorticity. I forgot to write it here. Okay. So this just says that vorticity rotates the order parameter. And this says that, for instance, shear flows also rotate the order parameter. In liquid crystal, this is called the alignment parameter of this lambda. Actually depends on molecular shape and says that, for instance, long tail molecules tend to align with the shear flows, essentially. And it's that alignment that leads to local order, and it is driven by activity because the flows are driven by activity, ultimately.
Thank you.
Okay. Any other questions?
And lambda, the sign is over positive?
For long thin rods, it's positive. For discoid, it will be negative. And if lambda in magnitude is bigger than 1, you get flow alignment. If lambda in magnitude is smaller than 1, then you don't have a steady state, you have tumbling. Yeah, that's right. Yes.
I have a question continuing on the
and I forgot to, David, you're supposed to remind me to repeat the question.
Okay.
Thank you. Okay, let's see. Yes.
But there is a phase, there's the P, which definitely varies.
Not yet. Here, yes, yes. So that is true. In fact, I think it's the next slide. So my total density is constant, but now I'm going to introduce a concentration field that will vary. Okay, that's absolutely right. Okay, all right. So now I actually need to describe the mixture. And so to describe the mixture, I couple the hydrodynamics to the dynamics of a phase field, essentially a concentration field here. For which I have written essentially the same equation I wrote before, with the only difference that it's also affected by the flow. And in addition, my two fields, my two fluid, one is a passive Newtonian fluid and the other one is an active fluid. And so I also need active stresses and capillary forces that are present here that describe the restoring forces, the passive restoring forces due to interfacial tension, interfacial curvature, to only be present in the active fluid. So I should have said this concentration field is, if you like, defined to be 0 in the Newtonian fluid, 1 in the passive fluid. And these are sort of the set of coupled equation. Mu is again the gradient of the free energy as I defined it before, that I need to use to describe the system. I've also included gravity here because the gravity was present in the experiments. So yes, the total density is constant, but this phi can vary. Now, there's another really important simplifying assumption here, which is there's a single flow velocity. So in some sense, I'm assuming there's single viscosity, a single flow velocity. So this is kind of the simplest model I can really write down to couple the original model B, as it's called, to flow, make it model H, and then to liquid crystalline degrees of freedom. Actually, to study the interface dynamics, so in this system, experimentally, they see very little diffusion that usually result in something called austral ripening. And so to study the interface dynamics, actually, we make an even stronger approximation. We neglect these diffusive currents altogether, so that our phi really becomes what's known in fluid dynamics as a color function. We just solve this equation using some software called OpenFOAM, essentially, to describe the dynamics of the interface. Now, if you do that, you find that so these are the results obtained from the numerics. These are the results from the experiments. And first of all, what is increasing here is the concentration of kinesin. Here is my activity parameter in magnitude of the activity parameter. And as you can see, the interfacial fluctuation become stronger and stronger with the increasing activity. There are no fluctuations for essentially very low activity. And these, of course, they become highly nonlinear, which is not something we're going to really be able to describe analytically. And the other thing, these arrows are supposed to point to the fact that it looks like these fluctuations are also traveling. And that's something we want to try to understand. Yeah.
Effectively, is the activity making the effective surface tension negative?
It could. So,
this question in relation to ask if there is a characteristic wave number that is actually selected at least at the onset.
Yes. So if you so there is a class of model that has been studied quite a bit, in which instead of coupling to the nematic order parameter, one assumes that what activity does phenomenologically is essentially to just have interfacial stresses. Okay, let's go back here. Okay, these capillary forces contain the same mu that has been removed from the right-hand side of that equation. And there'll be a stiffness in this mu, which in equilibrium has to be the same. There have been models that instead of coupling to Q, essentially say that the effect of activity is to change these capillary forces, allowing for what effectively is a negative interfacial tension, because the extensile forces can really kind of shear the interface and extend it, driving a negative interfacial tension. And that leads, that can actually lead to large interfacial fluctuations. And now it's getting a little bit, my answer is going to be a little bit technical. It's going to be, at the end, we're going to come to this, but, and it can also arrest the phase separation, okay? But it cannot do something which is really change the topology of the order parameter, which is also seen in experiments. And so we feel that in order to do that, it really is necessary the coupling to the liquid crystalline degrees of freedom.
What you can compute?
They actually compare very well without
parameter that you tuned the activity.
Yes. So, and that parameter, it's really, yeah. But
can do it with a constant activity?
Yes. Okay, wait a minute. Let's see a little bit where I'm going, and then you can ask me again. Okay. Okay, so so we wanted to try to go beyond just simulating these equations numerically and try to see how far we can get with some analytics in describing the fluctuations, at least to linear order, because this is what we're doing is really just to linear order.
So Cristina, just a point of information, on the previous slide, We supposed to see waves in the lower left.
Well, I'll show more convincing evidence of
They
ways.
they go in both directions.
Yes.
Okay, and then you eventually tell us what happens to it. You change the sign of the activity?
No, if change, I won't tell you what happens if you change the sign of the activity. Okay, so what we can do here is derive a linear equation for the dynamics of height fluctuation in the following way. Well, first of all, the rate of change of the height is simply the vertical component of the velocity. Remember, the bottom is active, the top is passive. And so what we can do is to essentially solve the two fluid problem with the right boundary conditions, with the jump in pressure Laplace equation essentially at the interface, with including the active stresses in the active fluid and no active stresses in the passive fluid. And if we do that, we end up with an equation which has the following structure. There are two terms here. The one in blue, which I call relaxation, is a passive term, and it is describes the relaxation of height fluctuation due to surface tension, and in this case to gravity. And I've written here in the case in which I've actually neglected viscous stresses, but we calculated in general, it just looks more complicated, and I didn't want to write it. The one in red is the active driving, which is complicated, but the thing I want to point out here is controlled by the active stress is actually controlled by shear flows generated, shear components of the active stress, so normal shear and regular plane shear flow. Remember, the sigma is proportional to the Q. And it's not just localized at the interface. It actually extends. There are contributions that extend throughout the bulk. And of course, this is not so you can think of this, if you have in thermal equilibrium, this would be thermal noise that drives the dynamic of the interfaces. Now it is active driving, which is essentially correlated in space and time. And of course, this is not a closed equation because sigma depends on the dynamics of Q. And we could try to close it, but instead of doing that, just on the basis of examining by calculating correlation function of Q, of this tensor Q, in both simulation and experiments, actually, we decided to do something simpler, to simply model the correlation of this active stress that enters in this expression I wrote before, as an exponential, essentially, both in space and time, with a correlation length and a correlation time, that we extract from both experiments and simulations.
Cristina, U is a K here is the same relaxation rate you had on the previous slide.
Yes, yes, and it's the passive relaxation. I mean, you could forget gravity, and this would be just surface tension.
Is there a typo? Should there be an H of K and Q multiplying?
Yes, thank you. Yes, there's a typo, right? Okay, and if we use this to calculate the height fluctuation, we get a form that is actually useful to compare with what you would get in equilibrium. So if that forcing term was just thermal noise, then this would be the form of my height fluctuation. This is called capillary length, which is determined, got to write it down, this is determined by gravity, essentially is the cutoff of the fluctuations at large scale due to gravity. And the strength of the fluctuation is simply determined by temperature. What happens here is that the strength of the fluctuation is determined by the spectrum of, this is really the spectrum of kinetic energy, where the kinetic energy is controlled by this active flow. So this is proportional to the strength, to my alpha square, if you like, to the strength of these of the active stresses. And in addition, there is this other factor that comes from the fact that the driving force is actually correlated in time. And the spectrum of kinetic energy in these active turbulent-like flows has been studied before. So one way you can understand this is that, so in equilibrium, you really have independent quadratic modes, and every mode contributes, there's equipartition, every mode contributes independently to the spectrum, and so there is this simple form. Here, activity gives you this scale-dependent kinetic energy spectrum. So equipartition no longer holds, and the spectrum is really determined by the form of this kinetic energy spectrum. And actually, if you were really to try to map it onto an effective temperature, you would actually need to say that this is an effective temperature, which is huge, 10 to the 11 Kelvin. The spectrum of energy fluctuations, as I said, has been studied before and has this characteristic form. So there is really a strong dependence on wave number, which is really controlled by which dissipation mechanism dominates in the system. Because these turbulent flows have these vortices, and in this case, when you have both viscosity and friction, the size of the vortice is essentially determined by the screening, the length scale that screens the flow. And actually, a very similar type of behavior for the kinetic energy spectrum is observed in polymer, in the so-called elastic turbulence of polymer mixtures, and has been studied quite a bit in the constant, which is also another example of a sort of turbulence, a low Reynolds number, as people call it. Now, if we use these to compare with experiments, we get qualitatively similar results for the wave vector dependence of the spectrum. Here, I've actually used an angle fluctuations. Although, of course, our scale, this is kind of what Maha was pointing to, we really don't know how to map our activity on concentration of kinesin. And so the comparison is really not quantitative. The scales are actually different. Although we do find that the root mean square fluctuation in both cases scale linearly. So in our case, scale linearly with our parameter alpha, and in the experiment scale almost linearly with the concentration of kinesin. So there is some correspondence between the two here.
Where is the capillary length on the spectrum?
So unfortunately, in these experiments, the capillary length and the viscous length are of the same order, about 60 micron. And that is a problem because it doesn't allow us to distinguish between the two. Yes. We take the screening length to actually be the thickness of the sample, the thickness of the cell, because if you're doing essentially a lubrication approximation, you would estimate that the friction would be of the order of the viscosity, 3D viscosity divided by the square of the thickness of the sample. Yes. These are 2D simulations. We are just simulating system in 2D. Yes. And these are 2D simulations. Yes. Yes.
Where is the active length?
Once you have a friction, the active length is not what controls the size of the vortices, but rather the viscous length controls the size of the vortices. And yes, but here we haven't really changed the friction. So yes.
And in the next slide, as you've changed, in essence, the position of the peak in the experiment seems to shift.
True.
It does not shift, and whereas in the simulations it does shift. Is that understood?
No. I'm not saying that this is just our first attempt to try to understand it. Okay, so, but let me tell you about these active surface waves that I alluded to before, which I think is kind of a more interesting possibly thing. So first of all, so I claim that these arrows showed surface waves. Well, there's actually a more convincing way of seeing it is here. These are again experiments. What this is, first of all, is a map in space, in horizontal and time of height fluctuation. And what is supposed to see is that there are correlation in space and time of height fluctuation. And this is actually the dynamical structure factor that shows these sort of side peaks indicating, no, it's sort of like the Brig-UN peaks when you do scattering from a fluid, indicates that we actually have traveling waves in this system. And this is from experiments.
But the spectra don't have the characteristic square root of K, K to the 3/2 of conventional?
No,
You're
so
fine.
that's
Okay.
right. So capillary wave, we know that, okay, capillary waves have this characteristic K to the 3 spectrum that David was alluding to. And generally capillary waves in fluids come from the interplay of surface tension and inertia. And you can understand them very simply by writing again this equation that relates the rate of change of the height to the vertical component of the velocity that drives the fluctuation. And then in the simplest incarnation of a Navier-Stokes equation, you have the rate of change of the velocity is controlled by pressure gradients, and the change in pressure at the interface is determined essentially by Laplace law. And if you combine these two equations, essentially you get a second order in time differential equation for the height that corresponds to the fact that you actually have traveling waves with this characteristic dispersion relation given here. And I've dropped gravity here. You can include it if you want. But in the system we're dealing with, really inertia is really negligible. And so how can we get traveling waves in the absence of inertia? And in order to understand this, we have actually tried to develop a really simple description of a minimal description of the interface dynamics in terms of just height fluctuation and fluctuation in the director field right at the interface. And so essentially we eliminate the flow in the bulk in favor of fluctuations just at the interface using a method that was used some time ago by Alan Bray and collaborator to describe the effect of shear flows actually on interface dynamics. And so essentially what you assume is that the phase field is kind of a step function with a certain thickness at the interface. And for the pneumatic director field, we take advantage of a fact that has been pointed out some time ago by the group of Julia Yeomans, which is essentially that gradients of active forces at the interface lead to anchoring. So there is kind of an active anchoring effects that tend to align the director field for extensile stresses parallel to the interface. So, and this is actually seen both in experiments and simulations. You have a very thin layer of active fluid, which really follows the interface. And so motivated by this, we assume that the director is aligned with the interface, and the system is disordered in the bulk, but pneumatic at the interface. And so when we do that, and I try to simplify here. So essentially, we obtain two coupled equations where the dynamics of height fluctuation, of course, decays. And again, here, this is supposed to be an H. Sorry, I made some typos. This is supposed to be H. So it decays at a rate which is determined by the passive relaxation, tension, surface tension. And active stresses proportional to the director field actually drive the fluctuations and tend to increase the height fluctuation and amplify them. On the other hand, the director field decays, and it's actually when the height field is suppressed, decays due to passive relaxation, it generates vertical flows. And these vertical flows turn the director field, and there is a feedback between the two that essentially leads to traveling waves. Another simple way to look at it is that whenever you have two coupled first-order differential equations, you are going to get waves. But what is important is that there is the relaxation rate of the two quantities are different. And so we call these waves are due to sort of a delayed feedback between interface fluctuation and orientational order that results in these traveling waves. The dispersion relation is somewhat complicated. These traveling waves, first of all, so what is showing here is the real and imaginary part of the dispersion relation. The blue is the imaginary part that corresponds to the traveling speed of the waves. And the waves only exist at above a finite wave vector. So, and the wavelength of the waves is essentially of the order of the surface tension divided by activity. And the wave speed grows with alpha, it grows with the active stress, making this very similar to analog, don't know what happened there, analog elastic waves that has been seen again in polymer solution and also in plasma systems, waves that are proportional to activity. And we can calculate the dynamical structure factor numerically and also get something very similar, qualitatively very similar to our simple model here. Yes.
I can understand the analysis of the wave that one sees in polymers and regular dynamics are bulk waves.
Yes, it's true. It's true. They're different. In that sense, this is just, I confess that the main reason I pointed this out is, so, you know, there is in active matter, there is this so-called active turbulence, which is a zero Reynolds number turbulence, and is controlled by equation that look very similar to the equation people write for polymers, where there is this elastic turbulence. But they're very different because in polymers, they're still driven from the outside, while here they're driven from the inside, and the scale of energy injection is sort of self-selected. But I gave a talk like this in front of Victor Steinberg recently, who was very unhappy that I didn't mention the analysis. This is why I mentioned it here. But I think you're right. I need to think about this more carefully.
And the aurora borealis is what we're seeing in the lower right.
Yeah. Have you ever seen it?
I have not.
Me neither. So, okay, but I would like to spend a minute telling you about the effect of these active stresses on phase separation and coarsening, even though this is something which sort of just starting to look at. So now you can also now go back to my numerical model, and actually in this case you want to put back a diffusion for the phase field, and try to calculate the phase diagram for in the presence of activity. And what you find is that activity suppresses, we call it suppression of phase separation, in the sense that there are regional parameters where the system will be phase separated in equilibrium, but it's not when you have activity. So these color curves are the boundary of the coexistence region as you increase activity. And so if, for instance, if you go to this blue point and you start with a bulk phase separated system and you let it evolve, it just goes to a homogeneous system. And only if you go down here, perhaps, then actually there are two things that happen. First of all, phase separation still happens, but it's arrested. It's sometimes what's called sometimes micro phase separation, because now there are finite region of a particular length scale. And notice also that the critical point also moves to the left, and this is kind of understandable because this region where phi naught is negative is the region where there is more passive than active fluid. And so active fluid is suppressed in the phase separation, so it's not surprising that the critical point will march to the left here.
Previously, I thought you had fluids distinguished by 0 and 1. Here we've switched.
Oh, sorry, this is -1 and 1. Two different people did this. And so they, yes, absolutely right. Yes. And this has actually been seen in a somewhat different system, which is a mixture of DNA condensates immersed in this active microtubule fluid by Alex Tayar, which is actually a very similar effect, which is this suppression of phase separation with the critical point actually shifting towards the passive fluid rich region, though we haven't really attempted any detailed comparison here.
Is it possible to use experiments to extract alpha? Because now you have the ability to see the characteristic scale. Yes,
and in fact we have done that, but I don't. I don't have, well, I might have a slide on it if I search for it, but so actually, so we have done two things. I'm way out of time. We have actually extracted alpha from the interfacial fluctuation spectrum, And also, I was not going to talk about this, but this active fluid, when it's in a thin cell, in the vertical cell, actually climbs the walls. There is a strong wetting effect that can actually turn into complete wetting. But we can also, using sort of simple model comparing to the experiments, we can also extract alpha independently from the height of the wetting layer. And these values actually agree with each other. That's what we have done, but yeah. So I'm just going to show you very qualitatively. So there is this, so there are two things that activity does. It suppresses phase separation and it arrests phase separation, okay? And so it really, one thing we want to ask is how it changes the coarsening. So the coarsening dynamics in binary liquid is relatively well understood. You can characterize the system in terms of a single length scale that grows in time. And there is this, these are snapshots of the system at different time, and they're supposed to be self-similar. And the size of the domains grows with an exponent of 1/3 when diffusion or Ostwald ripening dominates, of an exponent of 1 when flow dominates, and that vects these regions essentially. Now, here I just wanted to show you simulation and the experiments. What does activity do? Well, activity in this is really, so first of all, the blue curve is sort of a very low activity, not quite zero, otherwise we have no noise, but we can consider this a passive system. And we've actually gone a longer time, and we do see an exponent which is close to the 1/3 exponent, at least in this relatively short time region. Then you increase activity, and first of all, activity actually speeds up phase separation, coarsening. Perhaps not surprisingly, because activity generates flows, although you see these interfaces are much more strongly fluctuating than they would be. The droplet remains kind of spherical, but these interfaces are much more strongly fluctuating than they would be in equilibrium. And if you increase activity even further, oops, what happened? Let's try to go back. Okay. Maybe. Okay, if you increase activity even further, it actually arrests phase separation. And what you get is what we call an active emulsion. I don't know why my videos are doing that, but what you get an active emulsion, essentially a steady state where these droplets are continuously breaking up and fusing with each other. And there is actually a characteristic size, a characteristic length scale for the droplets that depends on activity.
And this is supposed to be like those condensates that you showed us at the beginning?
Well, who knows?
The phase diagram then would this be considered just not phase separated? I mean, the emulsion.
We consider it phase separated. We consider it phase separated. So in my phase diagram, I'm sorry about the videos, but in my phase diagram, here is phase separated, and it's always, we call it arrested phase separation, okay? Instead, so we call it micro phase separation as compared to bulk phase separation.
So is there some length scale?
Yes.
That decides whether it's homogeneous or separate phase separated?
Yes, yes, I'm going to show you in a second. And in experiments, by the way, they very much, although again, this is just something we're really starting to look at, they now using an horizontal cell, they see very much a similar kind of behavior. Here you have essentially zero activity. Here you have intermediate activity where the coarsening is faster and the droplets are still round. And here you have high activity where actually this non-monotonic behavior and this kind of, again, what we call an active emulsion with these droplets fusing and merging.
Psi is the average droplet size associated with this?
Yes, psi is what I call L in the experiments, is the average droplet size. And we are starting to at least estimate, you can actually kind of try to estimate the scaling of the droplet growth in by essentially, if you assume there is a single length scale whose rate of growth is controlled by various processes, in particular diffusion and flow, And then you can kind of just do dimensional estimates of the various processes, which I'm really not going to explain in detail here. And what you would find is various regimes here, but you can also, indeed, as kind of Bert was alluding to, you can also ask about the scaling with activity of the length scale of the droplet when the phase separation is arrested. And you find a different scaling with activity depending on the various regimes. And so we think this is interesting because by actually looking at the scaling with activity, you might be able to identify which mechanism for coarsening is dominating the dynamics, essentially. But we haven't really, and actually the experimentalists are really trying to do that, to look at the behavior of the scaling of the droplet size with activity or with kinesin concentration. So I think there is still a lot to do here to really understand the behavior. And as I said, also it's very interesting that these active fluids actually climb the walls and have almost like a fountain effect in some regime, kind of like superfluid helium. But I will stop here because I think I'm going definitely over. So basically, what I've been trying to tell you is that activity, so we saw yesterday activity can drive phase separation through motility, but it can also suppress phase separation essentially via mixing by active stresses and flow. Now, this is perhaps not so surprising. We know the shear flow suppresses phase separation, externally imposed shear flows. This has been studied quite a bit. And these are sort of internally generated shear flows that also can suppress it. There are a number of new phenomena that occur at the interface. We have very large fluctuations, traveling surface waves. And I didn't tell you about wetting and wall climbing, but that's also seen in the experiments. And we have this kind of active emulsion phase that we're trying to characterize a little bit more, and in particular to better quantify the coarsening. And in general, especially from an experimental point of view, the question is whether you can use activity as a new way of sculpting the morphology of interfaces. And question that David just asked is whether any of this has any role in biology, which I don't have an answer for. So thank you. I hope there might be some more questions.
So there were lots of questions during the talk, and Cristina has been very patient with us, but I think she'd be willing to entertain other questions. I'll bring up this microphone if you're on the right, and Suraj will do it on the left. Any other questions for today? I have one. Can you separate these length scales, this activity length scale and the capillary length scale? Is there some experimental trick for doing that?
So the length scale depends on activity. You can change it by changing kinesin concentration. So you can separate them. Yes, and that's actually what they are trying to do experimentally now. Okay. Capillary, unfortunately, in all the experiments, capillary length and other length scales seem to be comparable, and so that makes it actually more complicated for theorists to try to figure out what's happening.
And as you said, it's sort of like turbulence, but not really, in the sense that there's a low Reynolds number involved.
Yes.
But is there an inverse cascade? Can you think of it that way?
Yes, you can think of it. So yeah, that goes. I mean, that happens in bulk systems. Right. In this bulk system, there is this so-called active turbulence, which you can think of as an inverse cascade. The difference, though, is that when you study turbulence, usually you inject energy at a certain rate. The rate of energy injection is one of the parameters that you use to characterize the system. Here, and that controls some of the scales, the various scales, and so on. Here, the turbulence is almost like self-organized because the system organizes the turbulent flow and the vorticity through this local energy injection. But yes, there is an energy cascade from small scale to large scale, but there is actually the spectrum of energy, unlike, you know, in turbulence you have the Kolmogorov region of scaling. You
Right.
don't have that. It's kind of collapsed to a single peak.
And in fact, you're taking energy out at all scales because of the friction.
Yes, that's true.
Any other questions? If not, please come back tomorrow, another talk at 4:30.
There was a question here, but
Oh, sorry, just me.
So in the part of the phase separation where you showed these droplets, so the droplets were the active fluid and the surrounding was passive?
good question. Because I cheated slightly. In the experiments, oops, sorry. My videos don't work very well. So here, in these experiments, the light color is the active fluid and the droplets are passive, okay? In our simulation, if I remember correctly, oh, what am I doing? Went the wrong way, I'm not very good. In our experiments, the droplets are active and the surrounding fluid is passive, okay? So, and why did we do this? Historical. A graduate student that was doing the simulation chose this. Now, we don't think it makes, so experimentally they have done both. We don't think it makes a big difference other than for the fact that because the phase diagram is, the critical point is shifted. And so if you are in a comparable regional parameter, the behavior seems to be similar, seems to be the same.
And that changes where the flows are. Is it inside the droplet or is it?
It
between?
changes the location of the critical point, essentially. If you go here, okay. So phi naught equal to 0 is 50-50. Phi naught negative is more passive than active fluid, and phi naught positive is more active than passive fluid. And when activity is present, you see that the critical point, if you want to define as the maximum of this curve, tends to shift to the more passive fluid region, okay? But if you compare the behavior, say, here and to the behavior that the passive system would have there, it doesn't seem to qualitatively change the behavior to have one, compare the two cases. Yeah.
Thank you.
Yeah. So I apologize for not repeating the questions to the Zoom audience.
Sorry, I forgot.
I don't-
Yes.
There's a couple of questions on the Zoom. I'm not sure.
Oh.
You see it on the chat, maybe?
I don't see the chat because it was removed, but maybe if I get out.
Or they could email you, maybe.
Okay, I see the question.
Okay,
The
do you
active
want-
stress model. Okay, Srijit Santosh is asking, maybe I can
Read it out loud.
the active stress model using the Q tensor is traceless, and it doesn't exactly just model the force dipole. It also models a force perpendicular to the force dipole of the opposite effect. How important is to model active stress like this? I'm not sure why, because if I take a bunch of force dipoles and sum up all the force dipoles, I actually get exactly this active stress, and I don't think there's a force perpendicular. And I could show you that if you were
On
here.
Zoom. Why don't you move on to the next one?
The next one by Sungan Ro. The interface fluctuation was proportional to K and thus would be small in the most small K limit. What would be the cause of this suppression? Well, it has to do, oops, I think they left.
Okay.
I don't know.
All right.
It
So,
disappeared.
oh,
Okay, I don't think I answered, but it actually has to do with the behavior of this relaxation rate when you include both viscosity and friction. The passive one has actually a complicated wave vector dependence, and you have to really look at that to understand what's happening. In a passive system, gravity would make the small k go to a constant. But here, there is actually an additional contribution. I think the friction is dominating here and gives an additional contribution, but it's a little bit, you really can only see, I don't think I have a good answer in terms of the physics, but okay.
so on that note, let us thank Cristina once again for our second beautiful talk. And again, as I said earlier, there'll be a third, I'm sure, quite beautiful talk tomorrow at 4:30. So thank you all.